In the summer of 2023, scientific collaborators gather in the Oregon wilderness. In the midst of the tall trees, they're there to look for some of the smallest insects.
We'll see under the microscope what's going on, but I am already pretty excited. Oh, that one. This one here. That's a good one. This is the one I'm most excited about today. And it's a female. So, and it has dusky wings. Dusky wings is always good. So, and we will let it lay eggs. You have banana. Very good. And here we have a pseudo obscura or persimilis right here. It wants to go up into the line, so cool. All right, let's go to number 2.
Flies belonging to the family Drosophilidae, commonly referred to as fruit flies, are found throughout the entire planet in almost every imaginable ecosystem. In total, there are more than 4,300 known species of Drosophilids. Their diversity has made them a key model system for biologists, who use the flies to study genetics and evolution of various animal traits. However, relatively few species are used in scientific study, and these often come from lab-bred strains. This leaves the majority of the fly's genetic diversity untapped.
Oh, this is my spare bucket with extremely expensive equipment, and I'm picking one of these. Oh, that's a nice banana right here. So this is good quality, and I'm not going to overdo this, but I'm going to put a little bit of yeast on here, and then I smush it up. So I'm Thomas Werner. I'm professor of biology, and I work at Michigan Technological University in the Department of Biological Sciences, and I consider myself to be an entomologist who loves fruit flies and butterflies.
Thomas strives to create a resource that can be used by researchers, educators, and students, and one that will provide greater insight into the existing natural diversity of flies. He set out to complete the monumental task of making a 4-volume encyclopedia of all the fruit flies belonging to the family Drosophilidae in North America. Thomas has already completed encyclopedias for the Northeast and Midwest, where some 60 species exist, and the Southeast, which has about the same number of species. Now, he has moved on to the Northwest, where, for the second summer, he has undertaken a 6,000-mile, 5-week road trip to survey various sites and document the 80 species present in the region. Thomas's wife, Megan, and his children, Oscar, Oliver, and Natalia, all travel with him on his field trip. For 10 days, one of Thomas's collaborators, Dr. Bernard Kim, joins him. Bernard is a postdoctoral scholar in Dr. Dmitri Petrov's lab at Stanford University. A group of junior scientists from Dr. Petrov's lab have also joined to help collect flies.
So, we actually still gotta eat the watermelon.
Do you guys want some warm watermelon?
I haven't
have it in excess.
I haven't had any water.
It might be slightly alcoholic.
Okay, you don't need those. This banana bottle I can give you, it's a little drippy. You see how all the bananas, they kind of become juice? Your face.
I just bought this car last week.
Yeah, Rusty, we're going fruit fly collecting. You like that?
On the first day, Thomas and the team set out to put up fly baits at locations around the campsite.
Gets shade the whole day. So, do you guys need more mushrooms and more watermelon? Okay, so then you have everything stinky and rotty that you need. Sometimes within minutes you see the first flies already coming. If you want to go to the bottom of fruit fly research, it is right down there where this yellow juice is.
It is an unconventional project for a scientist at a major university who would normally focus on hypothesis-driven research. Thomas was a postdoctoral scholar with Sean Carroll, whose lab works on cutting-edge evolutionary developmental biology. He published many papers in the major scientific journals, won state and university mentoring and teaching awards, and had mentored 110 students in molecular biology.
It was very interesting, but it stopped fascinating me at some point during my tenure track time as assistant professor, then associate professor, and now I'm a full professor and I can basically do whatever I want. You can do this, and really nothing escapes because the ground shakes as soon as the net hits the ground. Right? So you can do this. So when I was a 10-year-old boy, I was really bored and I lived in East Germany. And my parents forced me to go to the garden every time because we had so many spies around, and it was safer to be in the garden and to grow your own food. And so I ended up living in the garden all the summers, and as a 10-year-old boy I was really bored, and I started collecting some cabbage whites, and then I started collecting some prettier butterflies which were very hard to catch. And one day one of them landed in my bedroom on the 9th floor in the satellite city, and I stood in front of the question, will I collect this one if I ever want to start a butterfly collection, or will I let it go and never think about biology again? Yep. Oh. Now we can come here. This was the most skittish ones. Okay, so now I think I have all of my Drosophilids. Do the same thing again. Can do this the whole day. Nothing is going to happen. Pick a nice vial. Okay. The lab bench is your left leg. Make sure that this trash is out of the way. Oh, that's not trash. That's a young tree. Sorry. Sorry, young tree. Okay. So. Make a nice lab bench, up, put this somewhere where it doesn't get stuck, and then you go up. And when you're done, you do this. You can sit again, have another coffee with the left hand, but now you have to kind of pull this and switch hands to the left hand, so nothing comes out. You can have another beer or what. But at some point, so you have it like this at some point with the left hand, then you find your cotton plug, kill a mosquito from your ankle, and then you put this in, tilt it sideways, and put the cotton plug sideways and check there's no channel. If there's a channel of air, like this stupid cotton plug, it's really, okay. So now, wow, holy cow. Here. So this looks like a Drosophila Suzuki-eye, but it's way too big. This would be a male, but way too big. This one looks really like a Suzuki-eye, but it's also too big. Something is not right. So maybe they are not Drosophilids. Maybe there's... And these little things, these little black things, you will see them all the time. Those are not Drosophilids. They don't look like a fruit fly. Don't ask me what a fruit fly looks like. It is just, I look at it and I know it is one or it's not. I can't explain it. Does anyone want to do the watermelon? Okay, I'll let you try. Because biology is a hands-on science. It's going to be awesome. You can go a little closer. Oh, nice. Yeah, like a pro. Really good. I'll let you do it so that I don't have too much to sort through tonight. Yeah, very good. And go in with the vial. You will have to pull out the vial, you have to go with the vial up, bang, 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 with the vial up, bang, 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 and then he jumps and he lands. Hammer them down with the left hand, like you have to bang on the net. So, yeah, that way, yeah. Kind of in, yep, and grab the cotton ball, insert sideways, yep, and that's it. Oh, look at you. Only disappointments in here. Okay, one Drosophilid. Yay, you got the Drosophilid right there. That's one. Yeah, the other one is a shit thing. Oh, sorry.
1-to-1 ratio. I'll
Yeah,
take it.
1-to-1 ratio. Very good. Ah, very nice. Awesome. So, you did your first fruit. And then they will be very surprised when I say that the ones in their kitchen, those are the ones that I don't need. I need the ones that are living out in the forest, and there are about 30 species just in the Huron Mountains. So they are very surprised that I don't want the fruit flies of their kitchen. Yeah, because that is usually Drosophila melanogaster, what people have in their kitchen. And I look for the species that are not Drosophila melanogaster mostly.
At every field site, Thomas sets up his mobile lab, where he can sort and image fruit flies. This setup requires an impressive amount of equipment that is normally found in the sensitive environment of the laboratory. Megan came up with the idea of using their camping breakfast tent as the lab building.
Normally it takes me 4 hours to put up the lab, from the first tent setting up to carrying everything in. Today I got help, that was very good. I probably saved 2 hours. And it works just like a regular lab, so there are no disadvantages of it. I can do anything in this lab I want to. I can image fruit flies, I can identify them, I can preserve them. I have a little fridge here, I can rear them. So this one is fly prep. This is a fly prep drawer, and there are etherizers, and there's everything you would need to dissect flies. There are forceps in here, there are etherizers, there is a whole tool case actually. Keep it clutter free, but this one is all of my dissection tools. And the cooler. So this baby goes on 36 watt. These flies need it cold, and there are samples in the back, and there are all the flies that I have seen, and this is the rarest fly. Like only 8 specimens have been found in 100 years, and I found 3 last year and 15 this year. So I doubled the world record of Drosophila flavopenicola, and there is a couple, a male and a female in here. It's just like being in the home lab, just the air is fresher, I would say.
Earlier in his academic career, Thomas had sought the mentorship of Professor John Janicki, an evolutionary ecologist from the University of Rochester.
And so John Janicki came to Michigan Tech, and we went to the fruit fly baits, and he did something that was remarkable, that actually changed my career. He bent down with his net and caught something and looked through the net and told me what species it is. And I thought, I want to be that kind of magician. I would say some of these light, medium-sized ones are probably Drosophila subquinaria. This little one, the little yellow one, that could be Drosophila neotestacea maybe. Then we have a biggish one that is kind of with dusky wings, that one down there. So we worked for 5 years on this fruit fly book and published it in 2017. So that's how I got into actually this encyclopedia. It was the first book. We only wanted to write 1 book. And when I was done, I said, John, I cannot stop. What do I do? And he said, let's write a second one. I give you my nicest collection vial.
When the flies arrive from the collection trips, Thomas sorts them and records the species that were found.
I like to sort them by site number. So here are the 5s. Here's a 1, here's a 2. We have to fill the etherizer first so that we can put flies to sleep. We take a little bit of ether. Not too much, just so that the paper towel gets a little stained. Bang them down, bang them in. Try not to get the food. Okay, some food came out. That's unavoidable, but I don't like when food comes out. Oh, it's flying around. What is this? Okay, this is Drosophila. Okay, this is Drosophila persimilis or Drosophila pseudo obscura, just as I said in the field. So I was pretty right about that one. Oops. Next is 1 watermelon. Yep, 1 Drosophilid. The rest is just garbage. Yeah, that's typical. These are mushroom flies. These are mushroom breeders. Most of this is trash, so all the trash goes now into the morgue. So Bernard sent me an email and he asked me if I could help him send him some flies. And I said yes. I generate a huge amount of trash, these flies that I only throw away. The cool thing is that he actually sequences them and tells me sometimes, hey, you missed a super rare species, you misidentified it. This was actually Drosophila colorata and not Drosophila robusta. So this is a real win-win situation.
And so after talking to him for a while and he was telling me about this trip, I asked him last year, like, hey, can I come along just for a few days in Oregon? Since that was the closest field site to California. And yeah, hang out with him for 3 days and I was just like, this, I mean, you've kind of seen it all.
So do you have yours right in here or?
Yeah, it was up there.
Okay. Are still coming up from the bait? I'm not sure. I'm not super Okay, time to wave them into the net. So.
Thomas images some of the collected flies in the field lab to create pictures for his Encyclopedia entries. Imaging involves a piece-by-piece dissection of an individual fly. The process requires precision and a keen eye to detail.
The fly nub is an extremely stinky liquid that attracts bears, so it has to be in a container that is actually within a container suspended by a piece of tape, and it starts really smelling. I'm going to pipette this in the inside, just in the cotton plug. The fall asleep chamber here. She's big. Here, why does this thing have a... This is so strange. This is such a strange fly. It has an interruption. It has a light interruption along the dorsal midline. That doesn't make any sense. You have to get a really good grip and then just pull it through the fly. Yeah, that went well. Wing number 1 goes in alcohol for now.
I feel like sometimes people, see, or a lot of, maybe most of the time, people see science and art as two like very distinct things, but working with Thomas, it feels like you're kind of watching a craftsman like do his thing rather than just like a very dry kind of very dry process that people kind of expect science to be.
I have almost a feeling that this is actually Drosophila colorata. This is weird.
But I feel like a lot of scientists kind of see their work the same way. It's like things that they're incredibly passionate about, because otherwise why would we get paid so little and work so hard to do this?
I will check the nose. That will tell me if it is colorata or not. It has a white cheek, it has to have a white cheek, and it has to have a very narrow nose with a furrow in it. Yes, beautiful colorata. 99.999% sure.
You know, ultimately, like, we all just want to make something beautiful. Even though, you know, in science you're kind of constrained by the rules of scientific analysis, but it's still very much a creative endeavor.
Okay. So now the head is coming off. Without ripping the guts out, I have to be very careful because otherwise the abdomen gets deformed. If you want to go up the academic ladder and you try to go up with what I'm doing, you're probably going to fall down. People are way too scared to do anything like this, what I'm doing, because they don't produce papers, there is no question to it, so there is no hypothesis here with what I'm doing, there is no scientific question, there is just my desire to provide a service to the community without getting much out of it, other than I publish another book within 5 years. But this is the kind of stuff people don't dare to do and don't have time to do. So it is way out of the box. And I do it anyway because I'm just passionate about it. And now I can do what other people call academic freedom. Okay, this is academic freedom. Do what you are meant to do and do it with happiness. Okay?
None of the junior scientists have previously done fieldwork with Thomas. For some, it has been their first experience camping.
Day 5 is when shit hit the fan. So yesterday I broke my tent, but we patched it up. And then my car battery has died, but we're gonna jump it. And then I broke my favorite pair of sunglasses, but that's okay. I just can't really drive without them. But, you know, life is good. We're catching flies.
Just very different from what I'm used to, and I don't know, sleeping on the ground, I don't know, the bathroom situation. It's a lot of small things. But we're rolling with the punches, being very flexible. And luckily I have Bernard and Hannah, because like they've done this before a ton of times.
What about James?
Well, James.
And now the way to tell that this is suboccidentalis, the only way to tell actually is when you pick a male. Okay, I go all the way, and there are recurved hairs. These hairs that are recurved, see that on the front leg? Only the front leg. And if the length of it is longer than the diameter of the foot, then it is suboccidentalis. If they are shorter, it's occidentalis, but you see them only in the south. I think, oh, Bernard is going to be so happy. Let's fill this vial for him very nicely. Some of them are flying away, but it's okay. Bernard has less work to do then.
Okay, these are the suboccidentales, and I don't see.
Yeah, I think it's maybe a female, probably.
You have been given a glorious
Suboccidentalis male.
Yes. Oh my God. Oh, suboccidentalis male, she says. I'm guided by feeling, like adrenaline shock. Oh, I have something new. Before I even know what it is, the adrenaline sets in, and then I start figuring out what it is. Take the bumping pad down, bump them down, take this off, insert them, and bang it onto that soft thing. Don't bang it on the bench, otherwise we will have to do it to trick to get the next etherizer because it's a glass bottle. Okay, so you put...
It's very fun to watch because he gets so excited about like all of them and so excited about the little details, and you could tell he knows like everything.
I have no idea what it is.
Has this ever happened to you before?
Rarely. And when it happened, it was a new species. Yeah,
After 5 days, part of the team heads home, while the remaining scientists continue on to their next collection site. They travel south to the redwood forests in California, where they hope to encounter other species of flies.
Oh, God. Should have a biohazard sticker on the top of the box.
When there is time, Thomas works on researching information for the encyclopedias.
Kind of like the year and the title of this paper and the date. Here is kind of my table of contents for the new book. And all of these notes of all of the papers I have read go into a notebook about this species. So every species has its own notebook. If I if I don't have pictures, then little placeholders are going to be put there, and that is perfectly okay. From January through May, I read approximately an average 150 papers a month. So there are 80 species in the book. That is about 80 months of reading and typesetting, including putting the color plates together, like these ones. If you, for instance, look at one of these flies, it takes me 1 hour to 2 hours to just clean one of those flies. So from the taking of the picture to having this in the quality to put it on the color plate, that is about a 2-hour job. 5 years. One whole book is about 5 years. That's a pretty good average right now. And I will work day and night and weekends also, about 60 to 80 hours a week, doing other stuff as well, of course. I have to teach.
Okay, here's another text where he's like, I collected 25 Drosophila borealis at a river next to our campsite in Tinkham, Washington. This was an extreme event for me. Saturday, July 16th. This was a collection morning to remember. Still under adrenaline shock. I guess that was the day he got 5 or 6 flavopinical in a single bottle.
Pretty large, also 5 different species. I don't know, you sequence them and send the data back to him or whatever he did.
Ooh, there's some interesting looking flies here.
Oh my God, don't move. Don't move. Holy cow. How did they get in here? What's the entrance? Okay. Now we are getting everything that we need to finish writing the book. Okay.
You're gonna love
this.
this.
Date?
Oh, right there. Look at that.
I'm going to take a picture of the room. Oh my God, is that big Drosophilids there?
There's a lot of flies.
Oh, this
Do you
is
need a
fun.
vial?
Yes.
Or maybe 2?
Well, probably. I'll start with one. Oh my God. Oh my God, it's Drosophilids. There was once above it, a dark black one above it in front of my net, so I got that too.
Oh.
It was like, oh, something is around my eyes. Oh my God, we need more. I need another net, please. Oh my God. Oh. My goodness, we have to come back in the afternoon. We have to come back here, this is awesome. Whoa. Okay, one more vial, please. Just throw it
Vial.
next to my feet. Just throw it next to me. Thank you. It feels like I can skip the next 2 hours of coffee. Like it feels like a double espresso. It's like, wow, what do I do? What do I do? Where do I go? Where's my stuff? I need to put this for breeding or something like this. What do I do? Do I sacrifice it or do I breed it? What if? You know, there's all these what ifs. What is the best way to do it? Oh, we've got Miranda. Look at this. Okay. Okay, 4 watermelon. 5.
There's one in there that looks really interesting.
Oh my god, don't tell me.
James, ready to collect again?
Yep.
8 months later, Bernard and his team are out of the forest. In the lab, they continue the work started in the field, extracting DNA from the flies and sequencing their genomes to create a fuller picture of evolutionary processes.
So a lot is known about the genetics and development and biology of one of the species, Drosophilids melanogaster, and we are trying to use that as a perch to get into patterns of evolution in that species and in other Drosophila in general. You know, if you're interested in evolution in the world, you don't want to be basing your understanding of the world on these very peculiar organisms that form good model species in the lab. Sareptitis elegans, Drosophila melanogaster, they pretty much all the same in that they can be raised in the lab on sugar. But the world is not like that, and so if you want to understand the range of natural histories and range of developments, you need to get beyond these few species. People like to help us see the diversity of life. People like Bernard were able to allow us to use genomics with wild-caught single fly samples and build full genomes and do a lot of really interesting work. So my hope is that technology will enable the more fruitful and thorough engagement between this natural history approach and more hypothesis-driven conceptual approaches that dominate biology these days.
What we're after is trying to understand sort of like the generalities of the evolutionary process. Not just what happens in one species, but what's common to a lot of different species, what might be common to all of the species at the genus or the family level, and how these things change and what are the factors that cause these things to change, like ecological differences. So for us, the more species that we can study and incorporate into our data set, the more power we have to learn about these very broad-scale patterns and processes.
It like harkens back to the biologists that we read about, like Linnaeus and Cuvier and whoever was just going out and doing everything themselves and finding all these species and just sort of using the literature to guide them. And he's now also combining it with sort of these more modern techniques of genomics and sequencing and stuff to be able to corroborate his identifications and contribute to this greater resource that we're trying to create here.
I did my PhD in a computational lab, and I feel like I picked up really important skills there, but I feel like I've always wanted to be a biologist, not just like a computational person. It's like living things that you've taken and sequenced yourself, and you know exactly where you got them from. And yeah, it just makes it feel a lot more real to me.
It's made apparent to me that how I'm actually spending my time feels equally as important as to what questions I'm asking. I want to be able to spend part of my time doing field work and part of my time doing lab work. Both of those activities I find a lot of joy in. So in that way it has changed what kind of science I want to do in the future, because I want to be able to incorporate both of those activities. Yeah.
When I'm done with North America, then I'm probably going to drop down dead, because what I have in front of me, the Southwest, is going to take me at least 10-15 years. Maybe I'm not dying after that, but I don't know. I would like to do Hawaii. Maybe someone helps me to do the Hawaiian fruit fly species. The problem is there are 1,000 of them, and I would need somebody who collects the flies for me, and then I just image 1,000 fruit fly species. They are extremely beautiful, they are extremely rare, and they are dying out, so someone needs to document them, I think. There's nothing about Drosophila that gets me excited, I would say. There is no feature on Drosophila that gets me excited. Some people are excited about soccer and not about basketball. And it's just a round ball and people hopping around and kicking it with the leg or with their hands. But people are they just pick something and they stick with it. For me it was butterflies, and I was making fun as a kid of everyone who even looked at beetles and found them pretty. Flies were totally out of my league. I would have never thought I would get interested in flies. And now here I am, and I'm more excited about a fruit fly than about a butterfly. I think it is just about human nature to hold on to something and to get obsessed about it without any particular reasons. I don't know, it's just, at least for me, it works that way. I don't know how for other people. And this is when people run out of funding, they then start washing their fly vials, which is really nice. Everything is satisfying. Every little thing I do here from morning to evening is really highly satisfying. If I would have to do it in the lab, it would be tedious, but here out in nature, I enjoy the surroundings. I just feel really lucky and happy that I get to be out here with my mobile fruit fly lab and do this. This is kind of super special. This is not tedious, this is just really super special that I can do this on a campground. I love it. I just love it so much.